Every experience we have, every touch, taste, sound, smell, and sight travels the pathways of our nervous system, making connections between neurons stronger or weaker, making us who we are.
So our guiding question that's interested me since I was a graduate student is the broad question of how does experience modify the brain? Those modifications are referred to as synaptic plasticity.
MIT's Dr. Mark Baer is the 2018 Beckman Argyros Award in Vision Research recipient for his work on synaptic plasticity in the brain and its role in a visual impairment called amblyopia, or lazy eye.
We built on the work of Hubel and Wiesel and the students of Hubel and Wiesel that had looked at the effect of early deprivation of quality vision in one eye. That's a paradigm called monocular deprivation. As a consequence of this altered visual experience, there is a loss of vision in one of those eyes, and that condition is called amblyopia.
Amblyopia affects roughly 4% of the world's population. Treatment involves patching of the good eye in children, which forces the lazy eye to improve at the expense of the good eye. The Baer Lab's fundamental neuroscience research has revealed the synaptic basis of amblyopia and patch therapy. Through this understanding of how synapses change, their work has suggested new ways to fix amblyopia even in adulthood.
His work is actually a wonderful example of how, starting with a highly fundamental question, it can move, the work over the years can really move to some aspects of translation.
Synaptic plasticity is the idea that the strength of connections between synapses can change based on whether they are strongly or weakly activated.
What do we know about the mechanisms that make synapses weaker? What do we know about the mechanisms that make synapses stronger, and how are those related? That's how we started.
Synapses strengthen through the process of long-term potentiation, or LTP, a phenomenon first seen in the hippocampus. By the early 1980s, researchers determined that LTP requires strong activation of what are known as NMDA receptors. As a postdoc in Wolfsinger's lab, Dr. Baer discovered that inhibiting these receptors in the visual cortex in vivo also prevented both the development of and recovery from amblyopia. What was known about LTP at the time couldn't account for these results. First, why would patching work? With the strong eye covered, inputs from the weak eye should not be strong enough to make improvements in vision. Second, if NMDA receptors only help to strengthen synaptic connections, why would their inhibition prevent the development of amblyopia? Baer recognized there was much more to learn about NMDA receptors and synaptic plasticity. As Dr. Baer explored this question experimentally, the BCM theory, a mathematical description of synaptic plasticity, was being developed by Dr. Leon Cooper, best known for his Nobel Prize-winning work in superconductivity. The theory's three assumptions gave Dr. Baer the inspiration he needed to move his exploration of synaptic plasticity forward.
I think that was a very satisfying collaboration because it was a real interaction between theory and experiment in which the theory led to experiments and the experiments led to further modifications of the theory.
And then he had the insight to say, if this theory is right and if synapses can get stronger, they have to be able to get weaker. They have to undergo what we call long-term depression of synaptic transmission, or LTD.
LTD offered a novel explanation for why vision deteriorates in amblyopia. Synapses in the visual cortex weaken in response to weak or untimely activation. LTD and its association with weak activation of NMDA receptors was proven experimentally in the early 90s in the hippocampus by Dr. Baer's first graduate student, Serena Dudek, and later in the visual cortex by his first postdoc, Alfredo Kirkwood.
We now know there are now many forms of LTD, and we know that they contribute to many different brain functions, including a number of brain diseases, from autism to Alzheimer's disease to addiction.
So if you understand the fundamental mechanisms like Mark has done throughout his career, that will always have a wider applicability because the same mechanisms it's just how the brain works and how it stores memories.
Another feature of the BCM theory intrigued Baer. The threshold level of synaptic activity to elicit LTP adjusted depending on the history of visual cortical activity.
Immediately, I could see how the sliding threshold solves my puzzle about patch therapy. Because when we set it up so that the strong eye isn't seeing and the seeing eye isn't strong, cortical activity falls. There's an adjustment in the properties of synaptic plasticity. So now it would, in theory, be possible to elicit synaptic potentiation under conditions that normally would not be successful.
Later experiments confirm that changes in the properties of NMDA receptors are a molecular basis for these shifting threshold levels.
Yeah, we admired him a lot because of his work with the NMDA receptors and its involvement in visual system plasticity early in life.
With the Beckman Argyros Award, Dr. Baer's body of work has been recognized as significantly advancing the field of vision research.
I am extremely pleased and proud to have won this award. Arnold Beckman has had an enormous impact on science. As an inventor, he introduced a lot of technologies that have made a lot of types of science possible. As an entrepreneur, he built a very successful business and made this technology widely available. And as a philanthropist, he has supported a lot of fundamental research and particularly supported the careers of junior scientists, postdocs, and early investigators. And so he's just really left an incredible mark on the field.
The Beckman Argyros Award in Vision Research was established in 2013 in honor of Dr. Beckman's friendship with philanthropist George Al Argyros. Argyros served as chairman of the board of the Arnold and Mabel Beckman Foundation for 22 years, during which time he oversaw $600 million in philanthropic endeavors. The award recognizes and supports the innovative work of outstanding scientists addressing challenges in vision research.
This type of award is enormously valuable because it gives you a little bit more freedom to do high-risk, high-reward science.
This will include exploration of a treatment for amblyopia based on principles Dr. Baer has discovered throughout his career. Inspired by previous research, he and his collaborators are rebooting the visual systems of subjects by silencing their retinas with injected tetrodotoxin.
So in that situation, where activity levels are low, there would be a shift in the modification threshold for synaptic plasticity, enabling weakened connections serving the impaired eye to recover.
That is within the framework of the BCM theory, so it's driven by that.
We use mice that have poor quality vision in one eye, which is reminiscent of human amblyopia. And what we're able to do is temporarily silence the retinas. Upon recovery of activity in the retina, the animals then also have a recovery of ability through the eye that previously had poor quality vision.
It was an amazing result. It's one of those, relatively few jaw-dropping results that one gets in a scientific career.
New studies supported in part by the Beckman Argyros Award will help further these findings, as well as allow Dr. Baer to continue his trek into the fundamentals of neuroscience.
What motivated us was trying to gain understanding of how manipulations of sensory experience modify synaptic connections and therefore behavior in the brain. The time eventually would become ripe when we had learned enough that we might be able to apply that knowledge to come up with novel approaches to amblyopia. And I think that time has arrived.